what it's like here.
Okay. And I think you might have a career in marine science?
Hopefully.
Yeah.
I'd like to.
Yeah, very good. Do you want me to show you what we do here in the Phyto?
Yes, please.
Phyto lab? Okay. So, Phytoplankton, do you know what phytoplankton are?
Not really.
Okay, they're single cell plants.
Yeah.
We live in the sea.
They're fishy.
Well, they are single cells, so imagine your body's full of, your body's made of trillions of cells, yeah?
Oh, yeah.
So these guys are just one
Cells
cell.
inside plankton.
No, they are plankton, they're cells, and those cells are actual plankton. That's what we call plankton. And we call it plankton because they just float in the sea, and that's why it's called plankton. Yeah, so I'll show you. So these are, each one is a cell, okay, and they're just moving around.
So they're all moving around by themselves?
They just move around by themselves. These guys is what we call dinoflagellates. Moving very fast now to look at them. Okay, so they're very, very small. These guys, each of them is about 15 micron. Okay, so
very small.
1 millimeter is 1,000 microns.
Oh.
So 15 microns is only 0.015 millimeters.
Oh, that's small.
Okay, so they're that small. So you can't really see them with the naked eye, so you need these instruments.
That's very strong microscope.
Yeah, this is an inverted microscope. Okay, so what it means is that the light comes from the top and your objectives are at the bottom, compared to, let's say, these microscopes here.
Yeah.
These are the ones you probably seen earlier where the light comes from the bottom, so they're called compound microscopes. Yeah. Okay, so we use inverted microscope for the simple reason that you can't really manipulate your cells in the compound microscope because the objectives are too close to the stage.
And new species in these?
Yeah, we have found a complete new species. So the next process is to name the species. Well, this could be another new species. So this could be the second discovery. Yeah, within probably a similar genus.
The grid and the squares are there. So you can see her. It's mapped out with the surface area.
Oh, yeah.
And what I have to do is I have to literally scan through each one of those, and any algae that passes my field of vision, I would identify and count it. So, and then overall we do a summary then of the entire batch to see what has cropped up the most and what has been in bloom or not in bloom.
Samples, all the cell counts and all the species identification, we put them in these reports and we publish the reports, which they go to the shellfish industry and to the legislators in Ireland. And in those reports we make some comments.
Is that safe? They're safe for not to eat?
Yes, we said there are no harmful species here, or there are harmful species here, and then that could trigger other type of testing, like chemistry testing. I don't know if you have seen that already, and then shellfish would have to come into the lab, and it has to be tested for toxins if there are phytoplankton toxic species in the water. Yeah.
Oh yeah. Improvements has this research led to in the Irish shellfish?
Well, what it has done, it has reassured customers that the shellfish that comes out of Ireland is of good quality, that is safe to eat, and that is monitored and quality assured by the Marine Institute's Biotoxin Shellfish program.